So I'm still on the first set of slides. I just wanted to wrap up kind of the intro to population dynamics, and then we'll switch over to the method slides. So part 3. So we talked about the types of data that you'll be thinking about are these intracortical neural recordings. And we talked about how you can think of the activity of populations of neurons is often constrained to lower dimensional subspaces or sometimes manifolds within the high dimensional space, but also that the way in which activity evolves in these spaces is not random, but it seems to follow stereotyped patterns, so rules to how activity there's clearly rules to how activity evolves over time, and that's what we're referring to when we talk about dynamics. And so I walked through this example of the pendulum just to give you some intuition of what we really mean by dynamics. So for a simple system like this, it's easy to think about where you have, you know what the current state is, the position and velocity of the pendulum, and you know that how the system evolves over time is purely a function of its current state. So if I know the position and velocity of the pendulum right now, and I know those rules, x dot equals f of x, so which is that vector field, then I can predict the next time step and the next time step subsequently. Okay. So how does that relate to neural activity? So let's take a look at some work. This is from Mark Churchland and John Cunningham when they were in Krishna Shenoy's lab, where they looked at activity during what's called a delayed reaching task. So we talked about this a little bit kind of in the discussion, where you might have a type of movement where you know what you're about to do, so you can prepare for that movement without actually making that movement. So in this case, the monkeys are trained in this task paradigm. So they're reaching with their arms. It's kind of a two-dimensional workspace, so they're trying to reach in these trajectories on the two-dimensional workspace, and the workspace has like virtual barriers. So they called this the maze task, to instruct the monkey not just to make simple kind of straight line reaches all the time, but they can instruct a variety of reaches by the monkey knowing that it has to avoid the barriers. So they were recording about 200 neurons from motor cortex, that's primary motor and premotor cortex. And so here's an example. This is like one reaching condition where you can see there's a target at the top. There's also barriers that block kind of the straight line path, so the monkey knows that it needs to kind of reach in a curved path to get the target. But it also knows, it's really well trained, that it sees the target, but it shouldn't actually make the movement until it gets this go cue. Okay, so that's why this is called a delayed reaching task. So the key idea here is that you can separate out the process of preparing to make a movement from the process of actually executing the movement. You can kind of study those two things separately. So in this condition, so here's condition 1, the monkey sees the target, it prepares, and then it receives a go cue, which might be like an auditory tone or another visual stimulus. And as soon as it receives the go cue, it actually reaches with its arm, in this case in a curved movement. So, we'll very quickly contrast with kind of classic ideas of what activity in motor cortex should look like during this type of reach. So this is, if you crack open a neuroscience textbook like Kandel and Schwartz, you'll definitely see, Georgopoulos, work from Georgopoulos and Andy Schwartz and others, training monkeys to make reaches. And so this is a plot showing they trained the monkey to make reaches in 8 different directions, and these are raster plots. If you're not familiar with the raster plot, that's like they're recording a single neuron, and each one of these rows is a different trial of the monkey making the same movement. And the different plots are just for reaches to different directions. So hopefully what you can see here is that when the monkey's kind of reaching up and to the left, that neuron is more active. And when the monkey's reaching like down and to the right, that neuron is less active. So in the motor world, this is where we get the idea of a tuning curve. So if you kind of plot like monkey reaches in all these different directions, plot the number of spikes you see against the direction that the monkey was reaching in. So reach angle on the x-axis, spikes on the y-axis. You typically get kind of this, the data points that you can fit with these curves like a sinusoid. And this is kind of the basic idea of a tuning curve. So the idea here is that this neuron might be tuned to prefer movements in a specific direction, and for movements away from that direction, this neuron will fire less. So that's, few decades old, some very classic ideas on what neurons might be doing in motor cortex. So yeah, if you take the top left angle, that's at the peak of the curve, bottom right, that's at the trough. And the basic idea here is just the neuron's firing rate is some function of the kinematics, like the reach angle, or the kinetics, meaning like forces that the monkey's generating, or that sort of thing. So that, that's kind of like an old way of thinking about the data, and there's certainly some truth to that. But when Mark and John were looking at this data, they, you took some, like when we were talking a little bit earlier about, machine learning and unsupervised machine learning, they took some very simple machine learning approaches to visualize these high-dimensional data. Remember, they're recording from about 200 neurons, okay? So in a given trial, you might get something like this. So on this axis is different neurons. On that axis is time. And here I've colored the data, by different trials. Different conditions. So like, green might be, I don't know, reaching like down. Red might be reaching up or something, and you'd see multiple repeated trials of the same condition. So the first thing they did with this type of data is construct what we call a PSTH, a parastimulus time histogram, which just means take all the trials from the same condition and just average the responses across them and maybe smooth the data a little bit. And then you get kind of these, what we see on the right here, which are for each neuron, you just have kind of its average firing rate for that condition. So green tells you, for condition number 3, what does each neuron do on average? Black tells you, condition number 2, what does each neuron do on average? Okay, so I kind of told you what, like, the classic literature suggests you should see during these reaching tasks. Let's see what they actually saw. So remember, this is a delayed reaching task. We'll just start with some simple trajectories. So let's say the monkey is reaching up. So this is what the monkey's arm actually did, the red trace. And we're looking at a single neuron. So here's what that neuron actually did. So you can see when the target comes on, this is the delay period. So the monkey knows what it's about to do, but it doesn't actually make the movement until it gets that go cue. You can see the activity kind of ramp up gradually. And then when the monkey gets the go cue, when it needs to actually initiate the movement, you see kind of this rapid change in the firing rate, which might be sort of triphasic here. Okay, so that's for reaches up. And here's another like simple reach, mostly straight, like to the right. And then we see kind of this other pattern for the same neuron, right? So during the delay period, activity kind of ramps down, but then around the time of the go cue, we see this like very rapid change, has multiple phases, etc. And you can do that for a lot of different reaching conditions. And if you do that for a bunch of them, this is what the neuron's response, this single neuron's response looks like for, I think in this case, maybe 16 or 30 different reaching conditions. And all I'm trying to get across here is that there's not a simple relationship. It's not just like, there's clearly an angle that this neuron prefers. The neuron's response is a little bit more complex than that, right? So, clearly, like, activity is kind of modulated for red in different ways than green, in different ways from black, but there's also, like, if you kind of stare at this, you might see that there's some underlying structure here, but maybe it's not as simple as, okay, if the monkey's reaching in a certain direction, the firing rate is just going to go up. Okay, but that's just one neuron, and they had 200 of them, right? So let's plot some other neurons. You start to see some other, like, complicated firing rates that are also different, right? They saw a variety of patterns. And a key point here, just like looking at these individual neurons, their responses are a little bit confusing. It's hard to make heads or tails of what these neurons are doing for these different reaches. But so, we talked about this idea of neural activity kind of largely living on these lower dimensional subspaces. We went through like a 3-dimensional example. Now we're talking about a population of 200 neurons, so it's a 200-dimensional neural state, but the concepts are very similar. And then we also talked about this idea of there being dynamics underlying the population activity. So y'all covered principal components analysis a little bit in some of the lectures, just the idea of like an unsupervised way of finding lower dimensional representations that capture a lot of the variance of the activity. So in this case, they're going to take the 200-dimensional activity and reduce it down to 6 dimensions, and then search for kind of patterns within that 6-dimensional space. So here's all the types of reaching that the monkey was doing while they recorded from about 200 neurons. And this is just like a 2D view of the 6-dimensional activity. But what they found was that they could kind of rotate this activity, look within that 6-dimensional space, and see kind of very clear structures underlying this activity. So you see kind of in the final plot here, if this would ever stop. Sorry, I don't know if okay, stopped. So in the final plot here, what we're seeing, so the little dots represent at the time, remember this is a delayed reaching test. The monkey prepares and then it executes. So the little dots represent like kind of a low dimensional, if you look at all the neurons what is the activity doing just before the monkey starts to reach? So in the green case, those are reaches, let's say down and to the right. Activity is kind of out here just before the monkey starts to reach. In the red case, so reaching up, activities over there on the right, just before the monkey starts to reach. But hopefully what you also see is there's a very clear pattern that just, if you know where activity was just before the monkey starts to reach, you can kind of predict what's going to happen next. Like there's this very clear kind of rotational relationship in the data. Does that make sense? So, all they've done is take the 200 neurons activity, find a reduced dimensional subspace, and they're specifically looking for these kind of rotational patterns. I think, so they're searching for them. The fact is that they kind of found them. Any questions on that? Yeah, so JPCA is, instead of just like kind of looking for, so they started by reducing the data down to 6 dimensions. So within 6 dimensions, you can imagine you can split it into like 3 planes, right? 3 pairs of dimensions. So JPCA is specifically trying to find planes in which the data over time look like they're rotational. Does that make sense? So like PCA doesn't consider time at all. JPCA is saying like, all right, look at subsequent time steps. Could I describe them by like a rotational, like a linear rotation?
Sorry, this may be the question.
Yeah.
Why, can you remind me why rotational happened?
I don't think that there was a specific justification early on for why they should look for rotational patterns, to be honest. I think there's some theoretical work saying that like rotational patterns might be kind of a substrate for pattern generation. Like when you move the arm, you actually have to create patterns of muscle activity in order to move the arm like to different locations. So that's kind of a pattern generation problem. You have to output, the brain has to output all of these different patterns of muscle activity. So, I think that at least the early theoretical work was that, when I say early, I'm just talking about like 2010, that having these types of rotations might give you a substrate to generate patterns off of, kind of like a Fourier transform. Like, Fourier transforms have like, you like take your patterns and break them into oscillations. A rotation is a type of oscillation. So if you had like rotations of different frequencies, you add them together and you can generate basically any pattern you want. That was kind of the theoretical justification. I'm not saying that that's firmly like what's actually happening here. That's at least why they said they were looking for them.
The metaphor that's helped me understand this is they use the record player analogy, which is essentially you can think of the neural activity on this rotational as like the turntable turning the record, and then the up and down movement of the grooves as being the pattern that's being taken.
Yeah, I think that's a different justification than the pattern generation analogy. So that's kind of where they are now with this. Maybe more accurate. I really don't know. Yeah. Well, so the pattern generation analogy says that the muscle activity itself should be the sum of sinusoids. So the rotations give you different sinusoids, add them together in the appropriate ways. So like set your initial phases right. It's like, a Fourier transform, you have an amplitude and you have a phase. So set that by the initial state of your system, run this, and you should generate the right pattern. The record player is saying like, okay, that muscle activity stuff is orthogonal to this. So it's a good question. I think it's still like unresolved. They were looking for them. I think the surprising thing here is that, here you have a monkey doing like it's making about 100 different types of reaches, and yet, the activity for this fairly like complicated set of reaches, like in 200 dimensional space, there are these patterns that describe a lot of the activity. And I can quantify that a little bit more. So again, the dots represent the initial state of the system. So what is the monkey doing? Like, where does the monkey prepare? It's like where are the neurons after the monkey's done preparing, basically. And the trajectories represent how activity evolves over time. And they see a couple different rotational planes. But the basic idea, hopefully now connecting back to what we were talking about earlier, is that this is like very similar in terms of, you have a system that seems to be following dynamics, where if you know what activity is doing at a certain point, and you know kind of the rules for how it updates over time, you can predict out into the future what activity is doing. Sorry, were there more questions? Make sense? Okay, so yeah, that's why we went through the pendulum example, because effectively we see kind of similar patterns in the data. In this case, like rotational. I don't like personally. The specific kind of like these are like swirly little rotational patterns, I think is less important than the idea that like we can take this system and we can learn the rules for how activity updates over time, and that's a pretty good description of what the neurons are doing. Like, you have this complex high-dimensional space, but there's a kind of simple set of rules that governs how the neural activity behaves. That makes sense. Yeah. I think this is just showing that these rotations kind of last for a little while. So this is like going out further in time. How does activity evolve for, I think about 600 milliseconds of the reach? You see these kind of like very consistent patterns, like a clear amount of, like there's clearly something consistent to what activity is doing, regardless of like the specifics of the movement. Just like if you know the initial state, you can highly predict how activity evolves over time. And they showed this with a few different monkeys and a few different tasks, kind of these consistent rotational patterns. In later work with people who were paralyzed, we saw the same types of patterns in people making finger movements or attempting to make finger movements. So this is clearly not just like a monkey thing, but and the other thing is like, remember this is like a 200 dimensional space, right? There are 200 neurons. But even just like these 2D projections of the data represent 30 to 50% of the overall variance of the data. So, when we were talking earlier about the dimensionality of data and are data low dimensional, this is kind of what we're getting at, that you could imagine a system where you have 200 neurons, the data are 200 dimensional. But here we find, a very, very simple representations, 2D capture a lot of what these neurons are doing. You know, a 2D plane for 200 neurons is a little surprising. Everybody on board with this, or does that make sense?
So I guess the data variance here represents the neural activity.
That's right.
Is it fair to say also like how both noisy trajectory is corresponds to like is somehow similar or equivalent to the data variance explained? For example, if you go to previous slides, like with all the green dots, there is some variation, green trajectories. So I guess all those variance observed in the original trajectory space.
Oh yeah, but there are a lot of different green reaches also, right? So we do expect like this and this to be different, right? Sorry, I should have mentioned the green color here just represents like, if you look at kind of the start of the movie where like you can just see a very clear color code from green to red. And so then they go back and color the reaches. But we do expect like, reaching like this should not be the same as reaching like that, should not be the same as reaching like that. Does that make sense? So I might not have gotten your question, I'm sorry.
Yeah, I guess it's more like I guess this makes sense because what we're just saying is we expect that different trajectories they will correspond to different types of populational neural activity.
Exactly.
I guess probably it's not in the original analysis, but yeah, probably we can come up with a way of characterizing all those trajectories in this like 2D dimensional, 3D. Maybe associate that with I guess the PC of the neuron population.
Could you go a little further with that?
I guess the current 30 to 50% variance is in neuron population.
Is neural space, yep.
But what about in the original trajectory space? I guess
like you mean these reaches right over here, like how much variance is there? Yeah, no, good. Okay, now I got you. Yeah, good question. I mean, you can think about it like in the limit, they're only reaching in a 2D workspace. So that should only be kind of two-dimensional if you just thought about maybe the position, or maybe if you took the two-dimensional velocities, that would capture the entire thing. So yeah, those data are like kind of two-dimensional in the limit, if that makes sense. Yeah. But I see. So maybe you're saying further, like if you just looked at the neural activity and said,
how can I predict that?
Yeah, how much of the variance of the neural activity is predictive of this? It's actually like a surprisingly small number, something like, I'd guess on the order of 5% or something like that. So remember when I was talking about kind of the older theories of what the neurons were doing, like they're representing the different behavioral variables. So by that theory, the neural activity should be like 100% of it should be represented by what's going on here, right? If all the neurons care about is like, what is the arm doing right now, then you should be able to predict their activity completely by this. But it's actually a very small fraction of their activity is predictable. Yeah, good question. Okay, so I'm going to switch gears a little bit, and now, hopefully, we've gone through a lot of background, a lot of different topics. Dive into how we can use some of these ideas. Oops, wrong lecture. 1 second. I'll stop talking, pull up the lecture, and then start talking again. Yeah. Okay. So now we're gonna talk more about how to actually use like machine learning methods to try to uncover these population dynamics on single trials. I'll tell you a little bit more about what I actually. Okay. So this is. There's basically one slide in the second set of lecture notes that are helpful here. Let me just skip to it. Okay, so just for a little bit of motivation, and hopefully, you're kind of seeing the theme that we're getting at. But we know that kind of the rich like sensory, motor, and cognitive functions that the brain performs arises from the collective activity of really vast populations of neurons. And so, an emerging approach, like I just told you about, is to use population analyses to try to understand, what these neurons are doing. So rather than thinking that the neurons just represent those external variables like behavior, let's probe the internal processes a little bit. So like what is the kind of network doing? And these types of like population analyses, that itself isn't anything new, but it used to be really hard to get enough neurons to even like approach this type of question. Like recording 200 neurons used to be really challenging. You know, when I showed you those earlier slides, you know, from the 80s, they were recording 1 neuron at a time, right? Those are like painstaking experiments. So if you wanted to look at a population, you kind of need some way of, like you can't record simultaneously a population of neurons. So you do something like assume that the monkey's behavior is really, really highly stereotyped and just take this neuron recorded, you know, during this recording session and, you know, add neurons recording during another recording session and another one, and then kind of like trial average them, put them all together, and then your analysis kind of combines these different data sets, these different sets of neurons. But of course, you know, hopefully everybody knows that this like is changing, these limitations. But you know, just to give it some more motivation, so our ability to record from neurons is our capacity is increasing dramatically. This is a plot from Ian Stevenson and Conrad Cording from 2011, and it's been updated since, where they looked at, you know, they just surveyed the literature and said, how many neurons are people reporting being able to simultaneously record, right? So, you know, in the 60s, recording 1 or 2 neurons was, you know, about the kind of gold standard, and that's like kind of steadily increasing. But look at this y-axis. That's a log scale. So what we're seeing is a linear trend on a log scale, which means an exponential increase in the number of neurons that we're able to record. And actually, if you look at like recent data, it's maybe even breaking off of that, right? Like with high density silicon, or maybe you can start to fold in like calcium imaging. We're really starting to get to some massive populations, thousands of neurons recorded simultaneously. And you know, even if you just take this kind of like linear trend, our capacity is doubling about every 6 years. So this is like kind of a key opportunity. We can combine like large scale neural recording with new machine learning methods to do high resolution single trial analyses, meaning we don't have to like average over many repeats of the same behavior in order to generate these you know, population data. So that means we can study neural processes that were previously challenging to study. So imagine you want to study something that isn't like a stereotyped behavior, complex behaviors, or different model systems. Like in mice, it might be hard to train a mouse to generate like the same, you know, movement over and over and over again. Or cases where the single trial variability is itself the thing that you want to study. Like you can imagine for a lot of cognitive processes, decision making or something like that, if you average across a bunch of repeats, you're actually averaging out the effects that maybe you want to study. Or, you know, one thing that I'm really driven by is improving brain-machine interfaces, you know, for assistive devices for people who are paralyzed. If you want somebody to be able to move the robotic arm, you can't ask them to try to move it 20 times and average activity across all those repeats, but you want to be able to move it on a moment-by-moment basis and millisecond timescale. So this is where like new machine learning methods, I think, are extremely important. Okay, so what I'll be telling you about now is some machine learning methods that we developed to uncover single trial population dynamics. I'll start in cases where activity is predictable and show you how we model those dynamics using a technique called latent factor analysis via dynamical systems, or Lfads. I'll tell you a little bit about when activity is unpredictable. So when we break away from this autonomous dynamics assumption. And if we have time, I'll tell you a little bit about applications to 2-photon calcium imaging data. And I'm sure we won't have time for me to get to applications to cognitive data. Okay, so we've already been over this idea that, you know, we can think about a neural population, we can look at its kind of state, we see there are lower dimensional representations that capture a lot of the variance of the data, and also there are stereotyped ways in which data evolve over time. We already talked about this, so I can skip through it really quickly, but a key point I mentioned when they record data, when they recorded the data for the task, it kind of looks like this, where you have neurons over time. Each one of these little blips is for that neuron, like when was it firing an action potential. And the first thing they did when they processed the data was trial average. And they did the dimensionality reduction analyses to find those rotational dynamics that we talked about. But the basic concept that they were trying to get to was that neural population activity during a delay period goes to an initial state, and then activity during movement execution can be predicted largely based on that initial state by learning these underlying rules to how activity evolves over time, those dynamics. And this is a simplified view. Remember, we're talking about like 200 dimensions, so like a two-dimensional plane is not going to capture all the complexity, but it's still, I think, interesting that these are kind of dominant patterns in the data. Okay, so we took this as a starting point, and we said, look, they had to average across multiple repeats of the same behavior in order to pull out these patterns. But if they're really a property of the data, then we should be able to see them on individual trials, so individual movements. So how are we going to do that? So this is a method that I developed, co-developed with David Susillo and other collaborators, but David is a faculty at Stanford and is now at Facebook, was at Google Brain. Okay, so here's a basic idea. Remember that we think activity can be described by these unobserved dynamics where you have an initial state and then you have rules to how activity evolves over time. So if that is truly the case, then to describe any piece of data here where I have neurons by time for a single trial, then we should be able to transform this data to estimate the initial state. We should be able to model the dynamics. So each individual trial can have a different initial state, but all the trials should have the same dynamics. We think this is like consistent across all these different behaviors. So if we can do those two things, like find a way to estimate the initial state and model dynamics, then we should be able to take the initial state, integrate over time, and draw out kind of the underlying trajectory for how the data are evolving. So hopefully y'all remember, 2 hours ago when we were talking about autoencoders, and we were talking about them in the case of images. So we find compressed representations using those multi-layer perceptrons. But again, just to refresh your memory, the concept behind an autoencoder is pretty simple. You have one network that takes the input, in this case Z, and outputs a lower dimensional representation, S. And you have another network that takes that representation, S, and tries to reconstruct the initial data. Right? And just to review, the encoder is finding a compressed representation, or the system finds a compressed representation that allows it to best reconstruct the data. And this compression forces the network to preserve things that are important while hopefully discarding features that are unimportant for describing the data, like variability that we might think of as noise. Okay? So, I mean, this is basically a schematic of what we were talking about for images earlier. So what's the key difference between the image data and the type of neural data that we're looking at now? Well, one of the key differences is time, right? Image data don't have any temporal structure to them. But our neural data does. In fact, I've been going on and on about dynamics. That's all about how does data evolve over time. So clearly we can't just ignore, or we don't want to ignore that temporal structure. We want to be able to model it. So we're using something called a sequential autoencoder. So it's just like autoencoders that I was telling you about this morning, but the difference is instead of like those multi-layer perceptron feed-forward networks, we're using, yeah, good? Okay. Again, this is work with David. So instead of using multi-layer perceptrons, we are using recurrent neural networks. So the idea is, we talked about deep feed-forward networks where you have, the output of this artificial neuron is connected to the next layer's artificial neurons. The output of that layer's neurons are connected to the next layer. In this case, you only have one layer of artificial neurons that feeds back onto itself. So they're recurrently connected neurons, so that's why they're called recurrent neural networks. That's the key difference between these networks that we're using and what we talked about this morning. It's just instead of having like a deep feed-forward network, we have one layer in which each artificial neuron connects to every other neuron recurrently. So we're using recurrent neural networks to take the data in and output a low-dimensional representation. We're taking another recurrent neural network to take that low-dimensional representation and try to regenerate the input data. Same autoencoding concept that we talked about earlier, only difference is that we're using recurrent networks instead of feed-forward multi-layer networks. Yeah, there isn't really a difference. So what you're saying is like kind of a feed-forward network in which the weights are kind of identical between each layers. That would be like a feed-forward network with tied weights. Yeah, actually, the way we train these recurrent neural networks is to do what's called unrolling them. So we take them and turn them into a feed-forward network in which the weights are identical. So yeah, it's identical. A key thing though is that we feed the input in one step at a time, right? So the first thing the network does is just receive the first time step of input, and then we let it update through recurrence, and then pass in the next time step, let it update, pass in the next time step. Make sense? So the two networks here are acting in like an autoencoding fashion. The first one is trying to output an initial state. And the second one is effectively trying to learn the rules that determine how activity updates over time. So it can only use that initial state, and it has to describe the entire rest of the trial. So a minor thing is we're outputting, instead of trying to approximate the spiking activity directly of all those individual neurons, we're outputting firing rates for each individual neuron. So that's what you see, those like traces are for each neuron. Here's an estimate of its firing rate as a function of time. And the cost function in this case is the Poisson likelihood of the data. So the likelihood of the spikes we actually observed given the firing rates that are output by the network. And this is also super minor is we know that, or we observe that these firing rates are typically lower dimensional than the full dimensional data. So we can force the network to only allow kind of a certain dimensionality for the firing rates themselves. So it's got to output 200 neurons worth of firing rates, but we can use kind of a bottleneck there to make sure that it only uses 10 dimensions or 40 dimensions or whatever we'd like. That's a kind of parameter that we can set. Does that all make sense here? Yeah. Good question. So let's ignore the encoder for a second. You can just treat it as like a black. I mean, honestly, you could, like, you don't even necessarily need to use an RNN. There are a lot of ways you could do that in encoder. But let's just take that other process. So you have the initial state output by the first network, and then this generator has to regenerate the entire time series based only on that initial state. So that initial state sets the activations of those artificial neurons. So that's kind of what's encoded in the initial state. And then the recurrent connections determine how they update over time. So you have the initial state of that network, and then those recurrent connections tell us, okay, on the next time step, how should their activity change? And then on the next time step, et cetera. So it only really gets kind of one time step of input, like that first initial time step, and it needs to generate the rest of the trial based only on the kind of rules that it's learned, those recurrent connections. Does that make sense? Okay. So that's why we're kind of hoping that that generator network is approximating those dynamics, those rules, using by learning the right recurrent connectivity. Yeah.
Just to be clear,
Yep.
every time you make the input for the encoder RN, it's like a timestamp, very first timestamp across trials, across neurons.
So the encoder basically, so let's just think of one trial for a second, right? The encoder, we kind of serially feed in each time step, and then the encoder sees the entire trial, and then the final state of that encoder is what's output as the initial condition. Does that make sense? Yeah. So that second network, the generator, doesn't get anything until the encoder's gone through the entire trial, and then can only output like one time step's worth of information. Yep. Yeah, right, exactly. So in this case, there are 200 neurons recorded. So, the vector of input is 200 dimensional, and we passed it in time step by time step. And then similarly, at the output, it's got to try to match that input, 200 dimensions, match it time step by time step. Make sense? Okay, so some quick connections to what we've been talking about kind of all day. We've been talking about like dynamical systems, and you learned about them with Anne as well. So recurrent neural networks are themselves nonlinear dynamical systems. So that's kind of why they're a good thing to use for this problem of approximating dynamics, because a recurrent neural network is a nonlinear dynamical system. And the RNN's recurrent weights are adjusted during model training to approximate the system's dynamics. So we talked today about, how to train an autoencoder. You can randomly initialize the weights. You compute a cost at the end, and then through backpropagation, you can figure out kind of the gradient of the cost with respect to each weight. And then you can use gradient descent to update each weight. So we're doing the exact same thing here. And in fact, as we were talking about earlier, Laura, this is, we unroll the neural network, the recurrent neural network, so it's effectively a feed-forward network, exactly as like we were talking about this morning. We can use backpropagation in the same way as we were using for those feed-forward networks. Okay, so what do we actually get when we do this? So I already showed you kind of Mark and John's rotational dynamics that they got from trial averaging the data and then using PCA and JPCA. Here's what the output of Lfads looks like for those very same data. So here, once again, you're seeing these like rotational dynamics, but now each individual trajectory represents an individual trial. So rather than averaging across 20 repeated movements, now we're showing for a single trial. So this is more than 2,000 trials across an hour and a half of the monkey making reaching movements. We're showing for a single trial what those dynamics look like. And I think an important point here is, even though we kind of knew that they had found kind of rotational patterns in the data, we didn't tell the network anything about what to look for. We just randomly initialized its weights and fed in all these single trials in a completely unsupervised way. We didn't label the data. We didn't say, this is from this condition, this is from a different condition. But the network, so this is what the network kind of discovered when just trying to describe the data in this autoencoding fashion. So it was a nice kind of confirmation. We knew that there should be rotational dynamics, but that's exactly what the network pulled out, but now on an individual trial basis. Yeah? Okay, so this is kind of like a nice visualization of the data, but is it like really meaningful for anything? Well, now we can think back to relating the data to behavior. So remember, up to this point, this has been completely unsupervised. We just fed in these spike trains, asked the network to try to model them using this like dynamical system, these recurrent neural networks. But because this is motor cortex, we can also decode what the arm was doing at any moment. Because as we've already said, there's some relationship between the neurons' firing rates and what the arm is doing. So here's an example of a single condition where the monkey's reaching like up and to the left, and these like faint lines are different reach trials. So if we used kind of previous approaches for decoding neural activity, So we could smooth using something called a Kalman filter, or we can use different dimensionality reduction approaches. This is something called Gaussian process factor analysis. And then we can do either of those things and try to decode the monkey's reaching. You see that we're kind of getting like the direction roughly right. Like we know that the monkey's reaching up and to the left. But the moment by moment details, like the fine details of what the arm is doing, are just completely wrong. And there's a huge amount of variability across those different trials. And so we can quantify that by saying, now of the behavior, how much of the variance, so how much of the 2D velocity are we accounting for by decoding? And it's about 70%. So it's not awful, but it's not amazing. Alternatively, we can take the data and first denoise it with Lfads, and then use the same kind of very simple linear decoding methods. So unsupervised modeling of the data to denoise it, and then linear estimation to decode it. And now you can see the trajectories we're getting are a lot more consistent with what the monkey's arm was actually doing, even though the network didn't know anything about the behavior. We can quantify that, and now we're around 90%. We're capturing about 90% of the behavioral variance. So there are a couple things here. One, this is, to me, this was kind of a confirmation that, we've been talking about this idea of state space and these low dimensional subspaces and everything. And a lot of times they're used to visualize data, which is super useful for building intuition about the data. But this is kind of saying it goes beyond visualization. This is saying that these descriptions of the data are like closely linked to behavior on a single trial basis and on millisecond timescales. So we're decoding here at like a 20 millisecond. We're decoding like what is the velocity of the arm every 20 milliseconds. And another minor point here is the numbers I'm talking about are for if you had the full population of 200 neurons. We can also just kind of randomly drop neurons and say like how well does the system do in modeling the data and decoding behavior. And just to make a long story short, you can see like if you have sort of between 25 and 50 neurons and Lfads, you could do as well or better than previous methods with the full population of 200 neurons. So this is a really powerful way of kind of extracting information from the population activity. All make sense? Good? Any questions? Bless you. So we talked about cases where activity is kind of, we have this initial state, activity during the reach is well described by, these kind of dynamic rules. But what about cases where that's not possible, right? Like if activity were completely predictable, neural circuits wouldn't be capable of doing anything interesting. We couldn't respond to inputs or anything. We'd just be kind of like in these loops doing the same thing over and over again. So that's clearly like it was a good assumption for the specific behavior that we're looking at, but we want techniques that can generalize beyond that. So let's go back to our state space view, where we've said, oftentimes activity is predictable and can be modeled by these autonomous dynamics. So what about cases when activity is not predictable? So like, we have some prediction based on the vector flow field, and then look, all of a sudden the activity kind of jumps out there. So from the kind of dynamical systems view, we can say that that represents a case where there's maybe been an input to the system. And so basically what we can say is this kind of failure, like our inability to predict the activity, might reflect some external influence on the system. So in this case, we need kind of a more general model. So for unpredictable activity, we think about it as being modeled by non-autonomous dynamics, meaning the update in the state is a function not only of the current state, but also of potentially these unobserved inputs coming into the system. So I'll just very briefly, go over some machine learning details. Actually, I probably won't spend that much time on this, but the key point is we need to take the model that I described earlier, and now we need to allow for the possibility that RNN that is trying to approximate dynamics can also receive inputs at any time step. And so we use some other RNNs to be able to infer, when is our autonomous system being unable to describe the data? And if that's the case, that probably means that some input has come in and perturbed the system. So let's allow this other RNN to kind of predict what the input should be. And in our original Nature Methods paper, we kind of showed this proof of principle that on data where there are unpredictable perturbations to the monkey's arm, Lfads could infer that they were happening, when they were happening, and roughly like the identity, meaning different inputs for a perturbation of the arm to the right or up or down or to the left or those sorts of things. So kind of a proof of concept. Questions on this? So maybe the last thing I'll try to cover is, as you can see, like our model is starting to get pretty complicated. We have kind of RNNs all over the place, and yeah, so it's getting complex. And one thing I'm not even talking about is that there are all sorts of hyperparameters that now require kind of careful tuning. So this is one of those the devil's in the details. Getting this stuff to work can require sitting there and tweaking various regularization parameters and getting things to work right. So for an example of what that means, we have these recurrent weights that are being learned in all these different RNNs. You can have what's called an L2 penalty, which is penalizing the magnitude of the L2 norm of those weights. I'm not going to go into details on that. There are other KL divergence penalties, but we didn't cover variational training, so I'm not going to go into that. Key point is properly doing this might, setting these hyperparameters might depend on the data set size, the number of neurons, the brain area, behavioral complexity, all sorts of things. And that makes it kind of hard for people to just off the shelf be able to use. I'll just mention, I can't go through all the details of these figures because I want to make sure you guys can ask some questions. Ignore kind of the precise labeling of these figures. Let me just tell you that on the x-axis here is the loss achieved by models, and on the y-axis is how well we can, when we evaluate the models using something like behavior, or how well do they describe the average responses, in this case average responses, that's what we actually get. So when we apply these models to different data sets and use different hyperparameters, the key thing you're seeing is that when we think the model is doing better, that's the x-axis, that doesn't necessarily mean it's actually doing better, right? So ideally here it would be better likelihood, so when the model optimizes better, it's better able to predict responses, but it turns out there's a lot of quirks to these models, and that's not actually the case. We had a NeurIPS paper kind of solving that. So we have a new technique called autoencoders, which is not actually in revision, it's now accepted in Nature Methods, where we basically found ways of getting past this problem, automating model training so that regardless of kind of the data set we apply to, now look at these purple dots, we don't have to tweak these hyperparameters manually. We're able to let the system just find solutions, and the system can find solutions that are just better than the previous method. So look at the y-axis here. In every case, the system outperforms the previous method. So now we have, so this outperforms Lfads in all brain areas and behaviors tested. So that's motor cortex, somatosensory cortex, dorsomedial frontal cortex, that's a cognitive area. And it's completely automated, meaning without knowing how the system works, you can kind of drop in data and just hit go and things work. And so over lunch we were talking about what's the utility of these deep learning methods, and I think there's a lot of just practical utility for having better tools to analyze your data. So, but if we make these tools so complicated that you need a PhD in machine learning to be able to use them, I don't think they're gonna kind of receive a broad application. So we've been working really hard to automate out a lot of that complexity. Yeah, not going to have too much time to go into some of these other things, but I'm happy to chat about them. So hopefully, in the methods, or sorry, in the demo coming up, y'all will be able to learn how to code up this basic sequential autoencoder. So that kind of first model that we talked about, working with some of the same data that we talked about, recordings from motor cortex. Yeah, looking forward to it. Thanks.